Okay, so welcome back from the break. Let's take a look at the concept check questions and let's see if we can figure out what's going on. So what are the chances of getting an ace from a 52 card deck of cards? Well, there are 4 aces in the deck, so the chance is for 1 ace. Oops. Let's see what color we've got. Yeah, let's do it. There are 4 aces in the deck. So it's 4 out of 52. And that's 1 out of 13. You can convert that to a fraction if you want to. An ace and a king. Well, here it's going to be a little bit different. So there are 4 aces in the deck. So again, it's 4 out of 52. But now once we take out one of the aces, we've only got 51 cards left. So it's not going to be 4 out of 52 again. It's going to be 4 out of 51. Now I sat down and calculated this. That equals 16 out of, is it 2,652, or approximately 1 out of 166, or if you'd like it as a decimal, 0.006. So about 0.6% chance of getting an ace and a king combination. The next one, 2 aces. So here it gets a little bit more difficult because for the first ace, it's obviously 4 out of 52. But for the second ace, we're down an ace, so there are only 3 aces left. And we're down to 52 cards. So that becomes, sorry, 51 cards, not 52 cards. So that now becomes 12 out of 2,652, which is 1 out of 221, or as a decimal, 0.0045. So the chances are even lower of getting 2 aces. But that's just simple statistics or probabilities. Now, let's take a look at some actual genetics problems. So I'm going to erase these so that I can do the genetics problems. So let's erase those. If you want the numbers, just scroll back, pause it, and copy everything off again, because it'll be saved in the recording, so you won't have lost that. Put the pen back in again. Okay, given the following cross, big A, little a, big B, big C, little c, crossed big A, big B, big C, little c. What's the chance of getting a big A, big B, big C, little c? So here's the shortcut to make this easier. So because Mendel says that homologous pairs of chromosomes behave independently, that's the basis for independent assortment, and meiosis reinforces that because of the behavior of the chromosomes in metaphase 1, that independent and random alignment. We don't need to puzzle over the A's and B's and C's together. We can just look at the A's. So when we look at the A's, what we wind up with is a simple Punnett square that looks like this. So we don't even need to draw that horrific
64 square Punnett square. There is no need for it. And we're asked for a big A combination. Well, that's that one. So that would be a quarter. Now, if we go to the B's, we could simply just replace the A's with B's and we would still get the same Punnett square. And we're looking at big B, little b. Well, that's 2 out of 4, or a half of them are big B, little b. Then we want big C, little c. Well, again, that's a half. And we want big A, big B, little b, big C, little c. We want that combination, so that means we multiply. So it would be, well, 1 times 1 times 1 is 1, so it's going to be 1 over something. 2 4s are 8, 2 8s are 16, so 1/16 or a quarter of them are going to be that combination. We don't even need to draw the Punnett square. Okay, so let's take a look at big A, little b, little c, little c. And let's do exactly the same thing again. Big A, little a, that's a half of them. Little b, well, that's the same as little a, that's a quarter. Little c, again, it's a quarter. And we're going to multiply. So 2 times 4 is 8. 4 8s are 32. So that would be 2 out of 64 would have that particular combination, or 1 out of 32. Now, there's a thunderstorm outside, so you may hear some rumbling in the background. Let's look at number 3. Maybe you're even ahead of me on this one now that you're starting to see how this works. So big A, little a, that's a half again. Big B, that's a quarter. Big C, little c, well, that's a half again. So we're going to multiply. 2 4s are 8, 2 8s are 16, so it comes out as 1/16 again. So 4 out of 64. Not a quarter of them, 4 out of 64. Is there actually an even simpler way of expressing this? And the answer is yes. If the combination is homozygous, then the fraction is always 1/4. If the combination is heterozygous, then it's always going to be a half. Once you've figured out what all the fractions are that you require, depending on the combination that you're looking at, multiply, you'll get your answer. And this works every time. And it really doesn't matter. It works with the monohybrid square, it works with the dihybrid square, it works with the trihybrid square. You can extend it to any size. Hey, you could even do a 23-way cross, which would be similar to humans, and you could still figure out the number. Because this is just simple statistics and probabilities built around what Mendel tells us about the behavior of the chromosome, about separate pairs of homologous chromosomes, and what meiosis reinforces about the behavior of those separate pairs of homologous chromosomes. We just simply take advantage of it to make it much simpler to solve what appear to be really complicated genetics problems. So hopefully you are able to follow along with that. And the review sheets that I have for this chapter give you some problems to sort out based on that idea. So let's move on and take a look at some other things. And so what we're going to look at in this next section are some inheritance patterns that are related to Mendelian genetics, but actually lie outside of the basic assumptions that Mendel made about the alleles and their behavior that led to the results that he had. because Mendel's description of the behavior of the alleles and the traits that they determine is a little bit too simple for what we now understand is really going on. So let's take a look at some examples that lie outside of Mendelian genetics. So the first is this whole notion of one trait being dominant over the other, and it really is all about traits. I know the textbook talks about dominant alleles and recessive alleles, and most biology professors do. And I will admit that up until about 15 years ago or thereabouts, I used to talk about dominant and recessive alleles. And then I sat in a seminar given by a geneticist who talked about the notion of dominant and recessive and realized that I had been teaching the wrong thing for many, many years. It's not the alleles that are dominant or recessive, it's the traits that they produce that are dominant and recessive. So you'll notice that I've tried to make a big point out of saying the allele for the dominant trait or the allele for the recessive trait. We're talking about dominant traits and recessive traits because alleles themselves are neither dominant nor recessive. It's the traits they produce. And this is what you need to understand in order to understand these deviations from Mendelian genetics. So the first example here is something called codominance. In codominance, both of the traits are expressed at equal levels. One trait is not dominant over the other. Neither trait is dominant over the other. They are equally expressed. And so in this circumstance, we're talking about another type of blood grouping other than the ABO. And we'll talk about that a little bit later, because that is a non-Mendelian trait as well. But this one is called MN, and it's all about the presence of two sugars, the M sugar and the N sugar, on the surface of your red blood cells. If you are homozygous for M, you have the M sugar. If you're homozygous for N, you have the N sugar. But if you're heterozygous, M and N, then you possess both sugars in equal amounts on your red blood cells. Neither is dominant, neither is recessive. They are equally expressed. So let me just write in here, both traits So that's the explanation. And of course, this lies outside of Mendelian genetics because Mendel said one trait is always dominant over the other. Here's a circumstance where neither trait is dominant over the other. We consider them to be codominant. They're equally expressed. So right there, we can see that there's a break in this notion of dominant versus recessive. Then the next idea, called incomplete dominance, really brings us back to our snapdragons again. Because here, we now finally have the explanation of the blending. The blending is not the blending of the alleles. The blending is the blending of the traits, the pigments. Because here, red is a red pigment. And unlike in Mendel's white peas,
white in snapdragons means a white pigment. So when you cross a red flower with a white flower, you have both the red and the white pigment present in the resulting offspring. So naturally, the red and the white pigments blend together and you get pink. But you haven't blended the alleles. The red allele and the white allele remain discrete. It's not the alleles that blend, it's the traits, the pigments that blend. So if the geneticists of Mendel's day had bothered to breed two of the pink flowers, they would have got this result. And you can see here that wherever you get the two red alleles, you get a red pigment. So this would be the homozygous equivalent. Wherever you get the two homozygous dominant, wherever you get the two white alleles, this would be the homozygous recessive equivalent. Here is your heterozygous equivalent, and they are the pink. But remember again, this is not dominant or recessive, this is incomplete dominance. The traits blend together to give a trait that is somewhere in the middle, a mixture of the two. This is the idea of incomplete dominance. The red does not completely mask the white, they blend together. And so under these circumstances, with incomplete dominance, you could never get a pure breeding pink flower. Doesn't exist. And the other thing that you need to be aware of, while the genotype ratio is 1 to 2 to 1, and phenotype ratio and this is very characteristic of incomplete dominance. So the 1 to 2 phenotype and genotype ratios, those are considered to be definitive of incomplete dominance. So if you ever have a genetics problem, either in my worksheets or in the dynamic study modules or wherever you might be testing yourself, and you see data that gives you a 1 to 2 to 1 genotype and phenotype ratio, you can be very confident that you're dealing with incomplete dominance. That's definitive, it's very characteristic of it. So again, we have to remember that it's not the alleles that are dominant or recessive, because they don't interact. They are on separate homologous chromosomes. When one is active, the other is equally active. It's really what they produce, the traits that they produce that lead to the notion of dominant or recessive. It's the traits that are dominant and recessive, not the alleles. So here's another situation. In humans, there's a condition called polydactyly where you have an extra digit on hands and feet. Now on the hand, it's up here next to the pinky finger. On the foot, it's next to the little toe. What you may not realize is that this is actually a dominant trait. Yet the vast majority of us only have 5 digits on their hands and feet. That's the recessive trait. And what it means is that the vast majority of the population are actually homozygous recessive for the number of digits on hands and feet. And that also flies in the face of Mendelian inheritance, because Mendel would argue that the dominant trait is the one that's most frequent in the population. But what Mendel didn't understand, and here's where perhaps the lack of communication between Mendel and Darwin really becomes significant, because if both of them had talked, all of these questions could have been answered. What we have to recognize is that natural selection will favor one trait over the other. So it can't necessarily eliminate a dominant trait, and there are good reasons for that, because you only need to have one
allele, so you could be either homozygous or heterozygous and show the dominant trait. So you can't specifically eliminate the dominant trait or the allele for the dominant trait, but you can reduce its appearance in the population by simply favoring the recessive trait. And that's what's happened here. So we have to remember that natural selection So the rule of thumb here is natural selection trumps genetics in this case. It favors 5 digits. We're not entirely sure why 5 digits, because some of the earliest fossils of terrestrial animals, terrestrial tetrapods, can have up to 10 digits per foot. But it drops down from 10 digits to smaller numbers until it zeroes in around 5. And we're not entirely sure why 5 is the favored form, but it is. And so that means individuals who are polydactyl are rare in the population, but not unusual. I've had several students who are identified as being polydactyl. But normally surgeons remove the extra digits shortly after birth because they become inconvenient. But I also know that in some cases they've been left, and I've seen, I haven't actually met an individual with the extra digits, but I've seen video of individuals with the extra digits. They turn out to be not terribly strong. They don't have the musculature and the nerve control over the digits, so they turn out to be more of a hindrance than a help, and that's why they're typically removed early on. Then we come to the ABO blood type. So one of the issues here is that Mendel would propose that there would be only 2 alleles for a gene. That is a very rare situation. There are usually multiple alleles, and in the case of the ABO blood group, there are 3 alleles. And so the alleles are big I A, big I B, and little I. So big I A and big I B are codominant. But both dominant to little I. So little I is the recessive allele. Big IA and big IB are considered to be codominant to each other, but are dominant to little I. And that's what accounts for the 4 different blood types that we see in humans. So here are the alleles, big IA, big IB, and little I. Big IA makes the A-type sugar, big IB makes the B-type sugar, little I, no sugar. So to be type A, you could either be homozygous for it or heterozygous, big IA, little I. You made the A-type sugar and you have blood type A. If you're homozygous, big IB, or heterozygous, big IB, little I, then you have type B. Here's where the codominance comes in. If you're heterozygous big IA, big IB, you make both sugars. And so you have blood type AB. Remember the MN system that we talked about earlier. If you are homozygous recessive, little I, then you're type O, and you make neither sugar. Now, why would this be important? I'm not sure whether I've got a slide on this one or not. I don't. So let me tell you why this is important. If you are type A, you make antibodies to B, and so you can receive A or O type blood. If you're type B, you make antibodies to A, and you can receive B or O. If you're type AB, you make no antibodies. And you can receive any type. And so you're actually referred to as the universal receiver. If you're type O, you make antibodies to A and B. And you can receive only O. But O is known as the universal donor because you don't have either sugars, so your blood can be given to any of the other blood types because you don't have the sugars, so it doesn't matter what antibodies they make, you don't have either sugar. So you are the universal donor. And type AB is the universal receiver. Now you might also be wondering about the plus and minus. So I know my blood type is O positive. That refers to something called the rhesus factor or RH factor. Rhesus because it was found in the rhesus monkeys and we happen to share that. And it's a simple Mendelian dominant recessive. So if you're homozygous or heterozygous for the dominant trait, for the positive, then you are RH positive. If you're homozygous recessive, then you're RH negative. So the true universal donor is O negative, and the true universal receiver is AB negative, if you want the full details. But bear in mind that all blood types are necessary because all have value, because we have all 4 blood types in the population. For some reason which we don't fully understand, type O tends to be the most frequent blood type in most populations, rating around 30%, maybe a little bit higher or a little bit lower depending on the ethnic groups involved. In some ethnic groups, O is very low. In other ethnic groups, O is very high. Other blood types are favored depending on the different ethnic groups. It gets really complicated. But the general rule of thumb, if you're able to donate blood and you're willing to donate blood, please do, because all blood types are welcome and every blood type has a use for medical purposes, either to save lives, to create medical treatments. There are many, many things that can be extracted from blood that have value. So if you can donate blood and you're willing to donate blood, please do. And remember, you can build up your escrow account. You can add to your escrow account, which will translate into extra credit points by donating blood at some point during the semester. You still have time for that. Then one of the other things that Mendel spoke about is that a single allele gives rise to a single phenotype. So the purple allele gives rise to the purple phenotype, the purple flower. That is not necessarily always the case. So in some human conditions that are genetically determined, and there are a number of different genetic conditions we could talk about, but in this class we talk about sickle cell anemia and cystic fibrosis quite a bit in terms of their genetics. It turns out that the presence of that allele, whether as a heterozygote or a homozygote, produces multiple phenotypes. We're only going to really address the homozygous situation because that's where the most significant effects come in. So if we think in terms of sickle cell, and I think there's a better explanation of the results of sickle cell a little bit later when we talk about it as a recessively inherited trait. But if we talk about sickle cell, let me just run through it for a minute because it's easy to illustrate what I'm talking about with this. So if you're homozygous for the sickle cell allele, you make a misfolded component of the hemoglobin. So when that is incorporated into the hemoglobin, and rather than the individual hemoglobin molecules being separate molecules, they link together in daisy chains of protein, which triggers the red blood cell to change from its normal disc shape to that characteristic sickle shape. Those cells can no longer carry oxygen as efficiently because of the hemoglobin, nor do they survive as long. Red blood cells usually survive around 4 weeks. These only survive about 2 or 3 weeks. Likewise, they cannot fit easily through the little blood vessels called the capillaries, which leads to reduced oxygen to the extremities and to the organs, which leads to tissue damage. And in the case of the brain, also leads to brain damage. The longevity or lack of longevity of the red blood cells leads to spleen damage because the spleen is where the red blood cells are removed. The fact that you have to create more blood cells because you turn them over faster means that you start to deplete your iron reserves, which means you become anemic. That's where the anemic part or the anemia part of the name comes from. And to top it all off, because of this accumulation of tissue and organ damage, the lifespan of the average sickle cell anemia sufferer is greatly reduced, typically somewhere around 55 to 65, depending on the degree of tissue and organ damage that accumulates. Or because you have to be homozygous for a single allele. Multiple phenotypes. And we can make the same statement about cystic fibrosis. In the interest of time, I'm going to move on, but we will take a little bit closer look at cystic fibrosis later on. But that's pleiotropy, where a single allele gives rise to multiple phenotypes. The flip side of that is that a single phenotype is the product of multiple genes at different locations, where the two genes interact with each other to produce the phenotype, and this is called epistasis. And the classic example of epistasis that we refer to is coat color in dogs, specifically Labradors in this case. And so the question is, can 2 black Labradors give rise to the 3 coat colors that are recognized in labs? Black, chocolate, and yellow. And yellow has a variety of shades associated with it. I used to have a purebred yellow Labrador, and she was on the darker side. She was up in here, on the darker side of the yellow labs. And we'll talk a little bit more about where that comes from. So what is the nature of this pattern? Well, the B gene determines how much of the pigment melatonin you make. Sorry, melanin, not melatonin, melanin. Melanin is the pigment. It's a dark brown, almost black pigment, and its accumulation in high amounts gives rise to the black coat color. The E gene determines how much of the pigment is deposited in the fur. So big B means make a lot of pigment. Big E means deposit it all in the fur. So any dog that inherits a big B in combination with a big E is going to be black, because the big B says make a lot of pigment, the big E says put it all in the fur. So here are your big B, big E combinations, the double dominant, lack and high deposition being the dominant traits. And so, as you might expect in Mendelian genetics, there are 9 double dominants. Remember, that's what we saw back when we were looking at Mendelian inheritance. Then, if it was Mendelian, we would expect to see 3 and 3 with 1 dominant and 1 recessive. Well, we do see 1 dominant and 1 recessive as a 3 in 16 outcome, and that's the chocolates. The chocolates come about because they are little b, which means make very little pigment, but they are big E. And the big E means even though you don't make a lot of pigment, you're going to deposit all of that in the fur, and that gives rise to the chocolate coat color. So here is the little b, big E combination, and you've got 3 of those, as you would expect. But you don't get the other dominant recessive combination, which would be a big B in combination with a little e, And the reason why is because that's where your yellows come in. And remember, yellows are a spectrum of color, not a single color. But they are all big B, make a lot of pigment, but they're little e, deposit very little of it in the fur. So any that is big B, little e in combination makes a lot of pigment, but only deposits a small amount of it in the fur. So these are the darker side of the yellow labs. This is where my lab fitted in. She looked very tiger-like because she had blotches of orange-brown fur against a lighter color, and we called her Tigger after the Winnie the Pooh character. The other combination is little b, little e, and these make very little pigment, don't deposit very much in the fur, and these are the very light end of the yellow spectrum. And so there are 4 of these that have the yellow coat color. So rather than being 9 to 3 to 3 to 1, it's 9 to 3 to 4, because it's not a single, each of these genes does not give rise to a single phenotype. It's both genes and the combinations, the alleles of those genes in concert that determine the final outcome, the final phenotype. So this is epistasis, where multiple genes give rise to a single phenotype. It's the flip side of pleiotropy. Then there are some characteristics that are the product of not just 2 genes, but multiple genes. It could be half a dozen genes, we don't know. But there is a simple way of figuring out whether that is the case. If the phenotypes occur as a continuous spectrum of phenotypes with 2 extremes and a broad distribution of various phenotypes in the middle, what we call a continuum of phenotype, then you can be very confident you're dealing with what is called polygenic inheritance, where multiple genes and multiple alleles of those genes are involved. Human skin color is a really great example of this. So you know you have extremes. You have very light skin and you have very dark skin. And here we're not talking about albinism. That's a completely different story altogether. And then you have a variety of skin colors ranging from very pale to very dark. And if you were to plot all of these out in terms of frequency in the population, what you would get is what is called a bell curve or a standard curve or a normal curve, which would look like this. At the 2 ends would be the 2 extremes. So in this case, it would be the pale skin color and the dark skin color. And all the other individuals' skin colors, that spectrum would lie in the middle here. Human height shows a similar distribution. And here, when we're talking about human height, we're talking about from about 4 feet 9 to about 6 feet 4 inches. And you know that there are people that are shorter than that and taller than that. But those are the product of other mutations, other genes in action. But normal human height ranges from about 4'9" through to about 6'4". And I've actually seen a picture. I saw it when I was much younger, and then I talked about this in one of my classes some years ago. In the next class meeting, one of the students brought in a book with the same picture in it, so it's out there, of boys from a British boarding school that were arranged into columns by height at 1-inch increments. And then a photographer stood on the top of a ladder and took a photo of the columns of boys. And if you connected the top of the columns together, you would be able to generate one of these normal curves or this bell curve, this continuum of distribution. So any feature in any organism that shows this continuum of organism, that's very characteristic of a polygenic inheritance pattern. And then the other thing that we always need to remember is that phenotype is not just a product of the genes that we possess. There is a whole discipline in biology that's arisen in the last 15 years or so, called epigenomics. Which looks at the effect of the environment So one of the long-standing arguments that people have had about who we become has been, are we the product of our genes versus are we the product of our environment? And people have gone backwards and forwards on this for many, many years. What we now recognize is that it's both. It's not just nature, our genes, it's not just nurture, our environment, it's a combination of the two. So it's not nature versus nurture, it's nature via nurture. It's our genes and our environment working together that will eventually determine who we become. And if you're not sure that that is the case, just look at any pair of identical twins that you might know. If you have known them for a long period of time, you will know that over time they have become distinct individuals, very separate from each other. And that's because each of them is experiencing the world a little bit differently, and that is reflected in who they become. And they can often become very, very different individuals as a consequence of their interaction with their environments, even though they are genetically identical. And this is where this whole notion of the effect of the environment really started to hit home in studying human genetics. Just the fact that when we look at identical twins, yes, they do start out as being genetically identical, but over time their environment ultimately determines who they eventually become. And they may still retain a lot of similarity, but there will be distinct differences as well. They will be their own unique individuals as a consequence of that. So here's an example from the plant kingdom. These are hydrangeas. This hydrangea is growing on acidic soil. And this is growing on neutral soil. But they are genetically identical.
And if that wasn't impressive enough, I actually have a picture elsewhere, I don't have it in the presentation, of caterpillars from the same species, actually, from the same species. And I believe in terms of experimental, they were eggs coming from the same mother. But the caterpillars growing on twigs took a form that looks exactly like twigs. Those that were feeding on leaves took a more fluffy bud-like appearance, yet these are genetically identical caterpillars coming from the same mother. And if you transfer them between food sources, they change their body form. It's the exposure to chemicals in the foods that they eat that determine their final physical appearance, not their genetics. This is the whole point behind epigenomics, looking at the role of the environment on determining gene expression. We refer these as multifactorial characteristics, where there is a role for both the genes and the environment in determining the final phenotype. So let's take another quick break and look at a couple of concept questions again. What is the basic difference between incomplete dominance and epistasis? If a man with blood type AB and a woman with type O have children, what blood types would you predict in their children, and what fraction would you expect for each type? We'll see you on the other side of the break.